series that you are undertaking. One of the topics to cover is the antigenicity and immunogenicity, building from the topics which you have learned previously. And my name is Professor Nyagol. I'm not sure when time will come so that we get to interact face to face. It will come, I don't know when. So for now, you had seen my face, so it's not necessary at this moment. You will be hearing my voice. Now, you must have defined the immune system as a defense mechanism capable of protecting the body against foreign intruder. So we'll be moving forward to understand what is that foreign intruder, and when it comes into contact with the body, of course, part of the immune response you must have learned, the two arms of the immune response which are activated, the innate arm of immune response, which is the immediate, and after some time, then there is activation of the adaptive arm of immune response. In both instances, what do they do to this foreign intruder which has come into contact with the body? One thing which you must have also learned keenly is that for the immune system to respond to this foreign intruder, which we will define clearly slightly later, there must be some specialized cells, and these cells must also express receptors on their surface. So the receptors which are expressed on the surface are the ones which are able to recognize the foreign intruder. So apart from the specialized cells with receptors on their surface, there is also the humoral portion of the immune response. And humoral portion, I just call it rudimentarily as humoral portion, because you must have learned it on adaptive arm of the immune responses, adaptive, specific, or how did you call it also? We have got adaptive or specific arm of immune response, separate from the innate arm of immune response. Now, what I'm talking about here is you have got cells which are involved in the recognition of that foreign intruder. But you also have the fluidic portion of the immune response, otherwise called humoral immune responses. And these humoral immune responses constitute the antibodies which are secreted into circulation. You also have serum proteins which are released into the interstitial tissue to help clear out that foreign intruder. You also have a group of about 30 proteins produced by the liver in response to, say, inflammatory processes, lumpsumly called the complement proteins. These you will understand later when you'll be dealing with that particular topic. So in reality, therefore, this foreign intruder defined as foreign to the body comes into contact with the body. And then it has to stimulate an immune response. How does it stimulate an immune response? It first must bind to the receptors of the cells which will recognize it. And then it stimulates the immune response. And in so binding to the receptors of the cells which will recognize it, we call it an antigen. Proceeding, therefore, to stimulate an immune system, we call it then an immunogen. Therefore, the two terms, antigenicity and immunogenicity. So this is what we are going to learn today. And what are your learning objectives at the end of this lecture? You should be able to differentiate between the antigenicity and immunogenicity in the context of that foreign intruder, which is able to bind to the receptors of the cell, or at least the cells have receptors to recognize that foreign intruder. And the recognition proceeds, therefore, to stimulate an immune response, therefore termed immunogenicity. So we shall also be going to know the factors that would influence the immunogenicity and differentiate the properties of B and T cell epitopes. When I talk about an epitope, is that portion of the foreign intruder, that portion of the antigen to which these B cell receptor and T cell receptors bind. I will elaborate much more on this aspect of epitope so that clarity comes when we reach at that point. However, one would be thinking at this point, why are we not talking about properties of, say, macrophages, properties of natural killer cells, properties of large family phagocytic cells? Now, when we talk about the cells which are key in clearing out the pathogen, or at least that foreign intruder from the body system, there are those cells which would be activated at the level of adaptive immune response. And this is why I kept on dwelling on the two arms of immune responses, the innate arm of immune response, which is not specific and is not able to clear out the pathogen. But then we have got the adaptive arm of immune response, which is geared towards now clearing out that pathogen. This is the reason which makes us focus on understanding the properties of the B cell and T cell epitopes, which means the antigens must be recognized by the B cell receptors, and also the antigen must be recognized by the T cell receptors. So we shall be comparing what is the difference between the recognition of antigen by the B cell receptors and recognition of antigen by the T cell receptors. And we go straight, therefore, in defining an antigen, which I mentioned to you is a foreign intruder. And I want to expand it slightly. When you talk about a foreign intruder coming into contact with the body, please do not focus your mind as in the medical field, you are only a quarter or 1/8 permitted to think in that direction, that that foreign intruder must be a biological antigen. In biological antigen, you are thinking in the context of infectious agents, be it bacterial infections, viral infections, parasitic or fungal infections. But I need you to expand the scope of your thinking beyond the biological agents, because any substance, anything which causes injury to a cell or a tissue would be classified as a foreign intruder. Hence, expanding from your thinking as a medical student of the biological injury to the body, you want to extend it to think also physical and also chemical injury, which can cause damage to the cells and tissues for which the immune system would be stimulated to respond. Hence, when you're talking about an antigen, just simply define it as a substance that can be recognized by the B cell receptor or T cell receptor. I want to repeat there. When you talk about an antigen, make it much more simpler than what you are seeing on the screen here. A substance that can be recognized by the B cell receptor or T cell receptor. Of course, here elaborated is that B cell receptors are those surface antibodies or immunoglobulins which are expressed on the surface of the B cell. Contrary to the T cells, which do not shed off, we have got B cells can express this surface immunoglobulins on its surface, but we also have the antibodies which can be released into circulation. Hence, you will be thinking about humoral arm of immune response, which you have covered by now. Now, you remember my third learning objective, which was given to you up, is going to help us compare these antigens which are recognized by the B cell receptor. How do they differ from the antigens recognized by the T cell receptor, in the context that for T cell receptor, there is a tripartite molecule which is required, the major histocompatibility complex molecule. We'll elaborate on that slightly later. Then I mentioned to you the first learning objective is that you must be able to differentiate between immunogenicity and antigenicity. And I mentioned to you, extending from the definition of a substance that can bind to B cell receptor or T cell receptor, which is the antigen, what is immunogenicity? Immunogenicity is therefore extending to stimulate an immune response. Antigenicity, just ability to combine with B cell receptor or T cell receptor. Immunogenicity is therefore ability of the immunogen or that antigen to induce an immune response. Once an antigen as a foreign intruder has been recognized by the B cell receptor or T cell receptor, it must stimulate an immune response to be defined as an immunogen. So therefore, you must know at this point that all immunogens must be antigens, but not all antigens are immunogens. If you are confused on that, you put for me a chat note so that I will elaborate further. Further terminologies to define in this topic are hapten. Now, sometimes immunogens or the antigens are focused on in immunology in the context of the vaccine design. So you have got, for instance, a bacterial infection which has got several strains, and you find out in these strains, given the virus, you have got COVID, which was in circulation just a few months back, and you think that the COVID circulating in Kenya is different from that circulating in South Africa or West Africa. Nonetheless, you have got conserved region in these strains of these COVID viruses, be it that one of China, Wuhan, China, or whatever, and you would want to use this conserved region among these strains of the COVID to design a vaccine. So this portion of the conserved region might not necessarily be immunogenic enough. And therefore, in the context of the vaccine design, this conserved region of the pathogens which might be used to design a vaccine is what is called a hapten, just a small molecule that is antigenic in itself, but may not be immunogenic. So this conserved region among the strains of the COVID could be used for a vaccine. Why? Because it is conserved among the strains which are circulating very cool. But is it able to induce an immune response? Not adequately. So it would be defined as an hapten. What do we do as scientists to make it more immunogenic, which can now go to the extent of using it as a vaccine design? There is a conjugate molecule called a carrier molecule, which is combined to this hapten to make it more immunogenic. So hapten in itself is a small molecule, can be antigenic, but it's not immunogenic. How do we make it more immunogenic? We combine it with a carrier molecule to make it more immunogenic. And additional terminologies which I want to bring you on board, not even on board comparison, we've talked about antigenicity, just ability to combine with the B cell receptor or T cell receptor. Immunogenicity is the ability of that antigen to induce an immune response. So define it as simple as such. I'm not sure what is written here because I don't read my notes. So you have got antigenicity, ability of an antigen to bind to B cell receptor or T cell receptor. Immunogenicity, ability of that antigen to proceed and induce an immune response. Quite simple as that. And I've mentioned that you could be having a conserved region among several strains of a pathogen, which could be used for vaccine design, and that is called a hapten, but it may not be immunogenic, although it is an antigen. So to make it immunogenic, you combine it with a carrier molecule to shift it from now not only hapten, but now to immunogens. And I mentioned to you, therefore, be it haptens, be it immunogens, all of them are antigens, but not all antigens are immunogens. And what is the role of that carrier molecule in the context of vaccine design? Talk about example, you have got COVID vaccine in circulation and you have got conserved region among several strains. So you have got a hapten here, the conserved region, which what is used to design a vaccine. And I've mentioned you have to combine it with a conjugate molecule, carrier molecule. So you have therefore hapten carrier conjugate, which if you now challenge a rabbit model or a mouse model with, you would find the immune system of this rabbit or the mouse model in an attempt to design a vaccine which can now reach clinical trials and be used in human beings. Antibodies in this rabbit model or mouse model would be produced which can react to the hapten. Antibodies would also be produced which can react to the carrier molecule. And antibodies can be produced which can react now to the conjugate, both the hapten and carrier molecule. This makes it much more important in the use of a carrier molecule to combine with a hapten to make a vaccine. When we proceed in subsequent notes, you'll be understanding now a shift completely in the clinical practice. This hapten now becomes that portion of the vaccine you want to use, and the carrier molecule now changes to be adjuvant, which is used in the design of a vaccine. Here we shall just elaborate briefly, but when you will be doing the concept of vaccinology, it will be much more clearer to you in the later times. So therefore, let's look at the factors which influence immunogenicity as our second learning objective. First, how much foreign in terms of evolutionary diversity is that molecule from your body cell. So foreignness defines the immunogenicity, how much foreign that biological or infectious agent is far wide apart evolutionary to your body. When it comes to contact with the body, it will be able to stimulate your body to respond in terms of immune response. You also have the type of molecule, and in this case, we talk about the chemical nature, in which case a protein is much more immunogenic than polysaccharide and much more immunogenic than lipid and much more immunogenic than nucleic acid. In the context of vaccine design, therefore, you'd be looking at the structural composition of that pathogen which you want to design a vaccine on. And if you can extract protein of that pathogen, then you would be sure you have a molecule which would be able to stimulate an immune response much more better than extracting the nucleic acid of that pathogen. Again, molecular science, which I've demonstrated to you in that hapten and carrier molecule, larger molecules tend to be more immunogenic than the smaller molecule, quite clear in the illustration which I've given you above. When you go to the composition, you typically know of sequences of amino acids. That is the basic structure of protein. And that basic structure of protein can move now to secondary structure of protein, then the tertiary structure of protein, then the quaternary structure of the protein. The coiled, much more coiled structure of the protein is the quaternary structure, which is much more complex. So heterogeneity would be able to increase immunogenicity in that the quaternary structure is much more immunogenic than the tertiary structure, than the secondary structure, than the primary structure, which is sequence of amino acids when you consider a protein. Also, you have learned about the phagocytosis and endpoint of phagocytosis, that the pathogen must be phagocytosed, degraded into small particles, then can be presented to the T cells to stimulate immune response. How best the body is able to degrade that pathogen would increase the immunogenicity. So you have got degradability as a factor which is able to increase the immunogenicity. Hence, coming back here to talking about the physical form, that denatured form after the phagocytosis is able to stimulate immune response much better than the native form of the protein, which has not been degraded. Now, if you look keenly how knowledge has progressed with time in terms of the vaccine development, we have got previously vaccines were being prepared using the whole organism, but now in the current market, you have got vaccines being prepared using just peptides of that microorganism. So in terms of increasing knowledge of vaccine development, things have improved from the whole organism to toxoid to subunit to peptides. Very cool. I like that because it makes your life very easy. What have you changed in doing that? You've decreased immunogenicity because the peptides, just like I've shown you here, that the molecular size tend to be more immunogenic than the larger molecules tend to be more immunogenic than smaller molecules. When you reach the peptide size of the pathogen, then you have decreased immunogenicity substantially compared to if you would have used the whole organisms. So in terms of decreasing immunogenicity, you've caused the problem by using peptides instead of the whole organism. But this is solved by having that peptide conjugated to a carrier molecule, which in the vaccine language we'll be talking about adjuvant at a later time. Yeah, but in terms of knowledge, yes, we appreciate that it is no longer important now to use a whole organism to design a vaccine rather than peptides, which are smaller molecules which are able to be sequenced and be handled in the laboratory. Other additional factors that influence the immunogenicity, the way you would respond, the way you're sitting there, if you get exposed to, say, influenza virus, COVID virus, and all that, the way you'll respond to it is different from the way I will respond to it. So genetic variability from one individual to another determines how you would respond to, or an individual would respond to foreign intruder, which in this context, largely we define as a biological agent. And also the dosage of the antigen which somebody is exposed to will be able to determine how best one can respond to optimal doses of those antigens. Also, number of doses of the antigen, in which case you may be called upon to administer boosters in patients who not only on trial, but even in vaccines which have got well establishment like the polio vaccine. You also have route of administration, which is able to determine how best the immune system will be stimulated. And you have the intravenous mode of introduction of a vaccine or biological agent would trigger much more of immune response than the subcutaneous, than the intraperitoneal, than the oral, than the inhaled mode. So vaccines have reached that are being administered currently mucosal, either oral or inhaled, but you must be sure that they do not stimulate the immune response as much as the intravenous injection of that vaccine. Excuse me, then I come to the core aspect of what we were talking about as the carrier molecule. There must be need to use an adjuvant in a case where you think that the component of the vaccine which you would want to use or which is in the market is not sufficient enough to stimulate an immune response. Previously, we called it a carrier molecule, but in the scientific and medical language, this is called an adjuvant, which must be mixed with an antigen. And when injected with it, it serves to enhance the immune response. And I've shown you how it serves to enhance the immune response. One, antibodies would be formed towards this adjuvant, antibodies would be formed towards the antigen, and antibodies would be formed towards the conjugate between the adjuvant and the antigen together. This I've shown you in the cartoon diagram in the previous slide. But how do these adjuvants work? One, they can prolong the persistence of the antigen, and in this way, they give the immune system more time to respond. Good. Point number two, they increase the size of the antigen by causing aggregation. This I've shown you. And they are also able to stimulate lymphocyte proliferation and also activation. They're also able to stimulate local inflammatory response and recruiting the phagocytic cells, typically the macrophages, to the site where it has been injected. And these macrophages, accumulation of them end up forming a giant cell called granuloma. So granuloma is typically aggregation of activated macrophages at the site where a stimuli has been recognized. Of course, at the site where you have injected in the body of a person that vaccine or that biological agent which you want to stimulate the immune system. The adjuvant is also able to enhance co-stimulatory molecules and co-stimulatory signals. I'm not sure if at this point I doubt you have learned the cytokines, but we have got co-stimulatory signals which could be recruited at a site where this biological agent has been injected or introduced into the body of an individual. And I don't want to go through this slide one by one, but some examples of commonly used adjuvants, which have been listed to you here, you can read at your own free time. Recently, there is typical use of bacterial lipopolysaccharides in the vaccine design as an adjuvant to stimulate the immune system much more better. So others which were previously used include alum and also the Friend's adjuvant, either incomplete or the complete Friend's adjuvant. This slide you can read at your own time. Now let's get to our point number 3, which is the third learning objective. Could we now start looking keenly at the differences between the site to which the antibody, which I mentioned to you is the B cell receptor, and the site to which the T cell receptor binds on the antigen? You remember me talking about an epitope, that portion of the antigen to which the B cell receptor or T cell receptor binds. So when you hear about the term epitope or antigenic determinant, these are the same two words, but the same thing. And is that portion of the antigen to which the B cell receptors or the antibodies on the surface of the B cell bind, or that portion of the antigen to which T cell receptors bind. And usually they differ, the epitopes or antigenic determinants to which the B cell receptors bind or to those which T cell receptors bind differ. So we'll be going to look keenly what is the difference in the B cell epitopes and what is the difference in the T cell epitopes, the T cell receptor epitopes and B cell receptor epitopes. And remember, I talked to you about the quaternary structure of a protein in our immunogenicity. Consider, for instance, there is somebody who is exposed to a virus, and this virus, for it to attach to the target cells, it uses these surface molecules called the glycoproteins. And these glycoproteins have got several portions to which the B cell receptor or T cell receptors can bind. And when exposed or degraded, then there would be exposure of those sites to which the B cell receptor or T cell receptors can bind, which I've defined to you are called epitopes. So we have got the glycoprotein here, which is not visible until it is exposed for you to see these several portions to which the B cell receptor or the T cell receptor can bind, otherwise called the epitopes. And each of these epitopes, like I get color, I'm not quite good with color, but if I talk about shapes, you have got the square, you have got water. So all these trigger different production of the B cell receptors to bind. And each of these will trigger the production of a B cell immunoglobulin, of course, or the surface immunoglobulin of the B cell to bind to it. We'll see that again in detail. I think there's a cartoon diagram coming ahead. But one thing you need to know is that each different antigen contains a number of different epitopes, and each of these epitopes trigger binding differently of the surface glycoproteins which are produced. So let's move to start understanding the properties of B cell epitopes. These B cell epitopes usually are dependent on the native. You remember me talking to you about the degraded structure is much more immunogenic than the native structure. Native, which means it is not degraded. And if it is not degraded, then we talk about either tertiary or quaternary conformational structure of the protein. So which means the protein is still folded. This is not making the B cell have difficulty in binding or identifying the epitopes. We will see contrary to this when we come down to the T cell epitopes. So the B cell epitopes are usually in native tertiary conformation or the quaternary conformation of the antigen, which means protein is still folded and B cell receptor can nonetheless still bind to those epitopes. But these epitopes must be accessible nonetheless. However, they are easily hydrophilic, which means they are water-liking and can be found in solution. And when I talk about solution, consider yourself having the fluidic portion or part of your body, the blood circulating in your body. Now, these antigens are usually found in the bloodstream. And in that context, it would be easier for the B cells to express receptors to bind to them. And these epitopes sometimes may be sequential, which means they are joined together, or they are non-sequential amino acids. This I will elaborate to you much better. When they are non-sequential, then they are termed as conformational epitopes. And again, B cell receptors will bind to soluble antigen, of course, the hydrophilic antigen, without necessity of third molecule. This term I must have mentioned to you earlier, major histocompatibility complex molecule. Now, let me elaborate slightly here before we move on for ease of understanding. The phagocytic cells, talk about, say, the dendritic cells, talk about even B cells are also the antigen presenting cell, but I need you to understand it later because today we are comparing something between B cell and T cell, so I don't want to introduce B cell here. I want to focus my explanation to the dendritic cell. Dendritic cell as a phagocytic cell, once it phagocytoses an antigen, degrades that antigen, and part of this antigen must be expressed on the surface of that dendritic cell for it to be recognized by the T cell. B cells do not need this kind of process to recognize the antigen because it has already its B cell receptors, immunoglobulin surface, immunoglobulins on its surface. So it doesn't require major histocompatibility complex molecule. In any case, the antigen presenting cells are not presenting antigen to B cells. They present antigen to T cells. So B cells will recognize these antigens without requirement of any major histocompatibility complex molecule. And usually I've mentioned that large antigens may contain multiple overlapping B cells, which can either be sequential or non-sequential. And we shall see what we term by sequential or non-sequential. But when we get that protein complex, which is folded, there's no need of denaturation of this protein for the B cells to recognize the epitopes. And I also mentioned to you that when we have what these surface immunoglobulins expressed on the B lymphocytes, they are able to recognize epitopes, and each of these epitopes would be recognized differently in terms of the B cells which would be formed on the surface. So we have got, say, the pink will be recognized by its own B cell receptor. The green will be recognized by its own, and also even which color is this looking like blue will be recognized by a different immunoglobulin. So on the surface of the B cell, there would be expression of the surface receptor, which means the surface immunoglobulins, which are able to recognize different epitopes depending on the conformational structure of those epitopes. And you see the right side, each of these epitopes has bound to its compatible B cell receptor based on the surface immunoglobulin, which is formed on the surface of the B cells. And I told you that then two kinds of B cell epitopes can occur. We can have the linear epitopes or the conformational, otherwise called discontinuous epitopes. Why do we call them linear and non-linear or discontinuous? When you get the epitopes piled together at one portion or in one region, pulled together, this is called continuous or linear epitopes. But when we get it scattered in the conformational structure of the protein, then that is what is called the discontinuous or the conformational epitopes. Now, I usually stress on this point here, but students have a problem understanding me. I hope you will understand me. Let me go slowly a little bit. You have got any viral hemorrhagic fever, and it is traced to Ebola virus, which is circulating in Uganda. Previously, you had got Ebola virus hitting DRC and other parts of the West Africa. That is good. Now, supposing you also have this Ebola spilling into Kenya, and I want to now make an imagination that a company has been engaged to design a kit for the detection of these Ebola viruses. And this same company designs a detection kit which can identify Ebola virus circulating in DRC. And this Ebola is different from the Ebola which is circulating in Uganda. And it's also different from Ebola which is circulating in Kenya. So let's move slowly. So you have got Ebola which is circulating in DRC is able to be detected in the epitope 1, which means that portion antibodies commercially can be designed to detect it. And then you as a medical doctor can sign out a report, oh, this patient has been diagnosed with Ebola, of course, that Ebola which is circulating in DRC. Now the same company can design also detection kit, which is detecting the Ebola virus which is circulating in Uganda. Nonetheless, the one circulating in Uganda can be detected in terms of expression of epitope 2. So the commercial kit containing the antibody can detect epitope 2. And that which is circulating in Kenya, God forbid, if it crosses here, the same company can say, look, the Ebola which is circulating in Kenya is different from these ones which are previously discovered in Uganda and that one which is discovered in DRC. So it designs a kit and says, look, this one, we can give you a kit which can detect it. And for that one, it can detect the Ebola based on epitope 3. So let's look at this linear or sequential antigens. We have talked about here linear epitopes which are formed here. So let's just come down here, look at this linear or sequential antigens. We have these antigens of the Ebola virus, which is well sequenced and could be traced to be having amino acids 1 up to 110. So if the epitope 1 has got the amino acid running from 1 up to 12, very nice. Epitope 2 is running from 8 up to 20, which means it is spilling into 1 and it is overlapping 3. Epitope 3 is starting from 20, and remember epitope 2 had reached 20. Epitope 3 is starting from 19 and running up to 33. Now, a patient travels from Uganda to Kenya, falls sick, gets admitted in Kenyatta, gets admitted in Aeropostale, gets admitted in where? The kit which is available in Kenya is that which can detect epitope 3. And you use that kit to test if this patient is Ebola positive. Tell me chances that you are going to sign out that report as a positive case. Where are they now? Who is going to sign out that report as a positive case? And why? Who is going to sign out that report as a false positive case and would want a repeat of the test? And why? Are we somewhere or the class is dead now? See, you told me you are around 300 students. I'm seeing only 276. Okay, let me assume they don't want to respond. The cat is just coming soon. It's no problem. So then let's go to the properties of T cell epitopes. And contrary to the properties of the B cell epitopes, I mentioned to you that B cell epitopes would be recognized just by surface immunoglobulins expressed on the surface of the B cells. However, for the T cells will require a tertiary molecule, which must be expressed by the antigen presenting cells. And I gave you example of a dendritic cell as a phagocytic cell, phagocytosing a pathogen and therefore degrades it. Part of it must be bound to a major histocompatibility complex molecule, which can now be presented to the T cell to be recognized by the T cell receptor. So the only thing is, which you must understand clearly, is that as contrary, as the same as the B cell receptor, the T cell receptors must also have the epitopes being accessible. But they tend not to be on the surface of the antigen, and therefore we term them as hydrophobic, which means water-hating. They are not quite soluble to be easily detected in the bloodstream. That was for the immunoglobulins or antibodies which are expressed on the surface of B cell. For T cell, the reaction would be confined much more on the lymph node, and I tend to think that you must have learned the structure or the classification of the immune system organs, the primary lymphoid tissues, secondary lymphoid tissues, quaternary lymphoid tissues, and you made a classification with a deep understanding on where the T cells really react when it comes into contact with the pathogen. So you have got these pathogens which could be internally hidden in the pathogen and not easy to detect, as contrary to the complex native structure of that antigens on the quaternary structure or the tertiary structure of the protein. And usually they do not bind to soluble antigen, I've explained to you, and they require processed antigen. In the case of the B cell receptor, it was a native antigen like that, which is recognized. But again, for the T cells, they recognize mostly proteins, but sometimes they would also be able to recognize lipids, glycoproteins, and this must be presented on major histocompatibility complex molecules before the antigen can be recognized. Now, this is the is called tertiary complex structure of the T cell receptor in recognition of the antigen. I need you to trace it from the T helper cell here, expressing the T cell receptor, which ordinarily ought to have just recognized this peptide, which is the antigen. But it can't recognize it until it is brought to it, bound to major histocompatibility complex molecule. In this case, I've given you example of class 2, major histocompatibility complex molecule, because the T cell recognizing it is the T helper cell. You will understand in depth when the major histocompatibility complex molecules will be taught to you as a topic. So you have got antigen presenting cell, having phagocytosed the antigen, digested it or processed it, and bind part of it to major histocompatibility complex molecule. That is when now the T cell receptor can be able to recognize. So this is the tertiary structure of the T cell receptor in recognition of the antigen. And as opposed to the B cells, you saw B cell was quite easy to recognize the antigen in circulation through the expression of the B cell receptor. X, a big X, because this is not a requirement for the T cell. T cells will not recognize antigen in this manner. In summary, one, we need to look at the definition of an epitope, which I've mentioned to you is called antigenic determinant, which is also the recognition site on the antigen, for example, antigen Y, antigen X, etc., etc. And if we look at the difference between B cells and T cells in terms of the recognition of the antigen, I mentioned to you for the B cells, it forms a bimolecular complex and it binds to soluble antigen. There's no major histocompatibility complex involvement. Antigen can be protein, polysaccharide, or lipid, and the epitope is nonetheless accessible and often conformational and hydrophilic, which means it's mobile and can be soluble and in circulation with the blood. For T cells, it forms a trimolecular complex, which I've just shown you. It is unable to bind soluble antigen, and the T cells has got major histocompatibility complex restriction, because without this, it will not be able to recognize that antigen. Antigen is always protein, contrary to the B cells, which bind to protein, polysaccharide, or lipid. And again, epitope is internal, which means must be exposed, must be denatured, must be linearized, and these epitopes are not easily soluble and found in circulation, but they will be found bound to antigen on the surface of antigen presenting cell and transported particularly to lymph node where the T cells are able to react to them. To that extent, we've covered all the learning objectives, defining antigenicity, immunogenicity, looking at factors which determine immunogenicity, and looking at the difference between the epitopes of the T cell and B cell receptors. Any question? I saw somebody raised a hand. Let me just go through the. I wanted to find out if somebody raised the hand to respond to before I come to these charts. If there's nobody who has raised the hand, I'm not sure these charts are so many. It is now enabled. Okay, yes, okay. Proceed. Not confused. Why confused? Kindly repeat, sir. It's too long. What do I repeat? Now it is clear. Okay, fine. False. False positive, or somebody's going to sign out that report as false. Somebody is going to sign it as false positive. Somebody is saying it's going to be negative. I don't know the one from Uganda. It is a positive since there is an overlap. There is a possibility of cross reactivity. Okay, negative because the one in DRC has epitope. No, we were comparing the one in Uganda and one in Kenya. Somebody is going to sign it as false negative. Like hujafanya fiti juu. Okay, false negative, but spelling to epitope 3. So is there no way to detect it? I don't know. You're asking the same question I've asked you, so I can't give you an answer. Sorry, it's false negative. It will be positive. I love this. Sorry, I'll inform him when he processes. Huh? What do you mean? Will not force cutting. Is almost finishing. Atarudi tu. Okay. Last slide is here. Hi. Yes, it will be positive because of overlapping of epitopes. Okay, I love this. Can this form our cut? Any other question? I assume silence means no question, no need for clarification on any slide, so we can end the lecture to that.
Students are just asking for clarification whether the answer is false, negative, or positive.
This is what I've asked you, and you see, I love that. Who is on phone? Who is talking? Introduce yourself.
Oh, this is Kelsey, the class rep.
So that's the same question I've asked you. So if you're asking me for the answer, then it means you notice. I mean, but I like it. You see how the class has got a variation of answers, which I really love. So it means these are independently thinking students. So I love this. So if you want to sit somewhere and say, look, This report is being done in the laboratory and I'm the one sitting here to sign it out. You, what would you have done? You say your name is?
Chelsea.
Alice.
Chelsea.
Chelsea. Anyway, carry on. So what would you have done? How would you have responded to this? Let me just go back to that slide, if you don't mind. See, we are here. So what would you have done? This is my Ugandan resident visiting Kenya, and there is an outbreak of Ebola in Uganda, and maybe Kenya is affected. And so they're circulating, just like coronavirus was in circulation. You get that strain of Wuhan, you get that strain in South Africa, you get that strain in Kenya, you get that strain which was ravaging Italy. So, I mean, you have got a Ugandan COVID, and with the kit, depending on the circulating strain there, could be detected using epitope 2. That one of Kenya can be detected using epitope 3. Now, when this patient is wheeled to the hospital under emergency, you want to move very quickly and decide to order for the test to be done. So you say, okay, fine. The kit which we have here in Kenya is that one which is detecting epitope 3. This is a Ugandan with the epitope 2. So then let's make a rational decision here, because we need to move quickly with the management of this patient. Chelsea, I'm still talking to you.
Oh, yes, yes.
I need you to sign out this report, because we are waiting for you in the casualty there, because we want to institute management.
Oh, no, I was just speaking on behalf of the students in the chat.
Yes, I don't refuse, but now I'm with you. I will also get another student.
False negative.
You'll sign it as false negative. Give me your reason.
Because there's no overlap.
What do you mean? But I see the epitope number 2 is running up to 20, and epitope number 3 is peaking from 19.
Oh, okay. Sorry, I misunderstood the diagram.
Somebody is saying it will be positive because of overlapping epitopes. We need the answer to that question, Prof. Please clarify on hapten. Hapten is quite clear up, so when you get these notes, it will just be visible to you. Do T cells attach to specific measure histocompatibility? Yes, T helper cells will attach only to measure histocompatibility complex class 2. T cytotoxic cells will attach to antigens presented bound to measure histocompatibility complex class 1. But I've mentioned to you that you will get clarity of this when you will be taught the measure histocompatibility complex molecules. What about co-stimulatory signals? Any example? Cytokines. Cytokines, you have got several interleukins, and this will also come clearer to you when you'll be doing the cytokines as a topic. Haron haptens antigen, which is not immunogenic. Yes, Njoroge, carry on. Njoroge, you raised your hand, carry on. We are having 5 minutes to explore. Haron hapten. Yes, carry on.
Hello.
Yes, carry on.
So I'm saying.
Yes.
So I'm saying because. Okay, because what we are looking at is just recognizing the Ebola antigen, and the epitope 3 is actually present in the Ebola antigen. As long as we recognize epitope 3, it means we've already recognized that this guy has Ebola antigen. So I think it will be positive.
Okay. I mean, I'm worried because you are only going to use this kit which overlaps with only one amino acid. Please clarify on the question.
Yeah, but my opinion was that
Carry on. There's an overlap. They were comparing the one in
My opinion was that because what we are looking for,
DNP and bovine serum albumin.
so can I carry on?
Yes, carry on. I'm getting.
Also, I'm saying my opinion is because we are looking for Ebola antigens, and epitope 3 is still present on the Ebola antigen. As long as we recognize epitope 3 and it is unique to only Ebola, I think I can give a positive for Ebola. That's my opinion.
Okay.
Unless maybe I didn't understand the concept of epitopes.
Okay, and false negative because it does not overlap. False negative, false negative. The kit in Kenya do not detect the epitope 2. Negative as false negative is positive, but the kit will not detect. False positive, positive. This I love. Okay, fine. So I'll release the class for now. So we explore this when the cut and the exam will come. You have a good morning.
Thank you.